Before diving into the explanation of how the wing disc develops, I'd like to introduce this technique, which you already know for sure, but well, it's important to understand the experiments that follow. Clonal analysis is just the study of cells that are clonally derived from a single cell and that are genetically identical, although they might be different from the surrounding tissue, which means that it's the study of mosaic tissues. And this is the genetic system that is usually used to obtain these mosaics. It is based on GAL4, which is a transcriptional activator that comes from yeast and that binds to the UAS sequence, which stands for upstream activating sequence. So this GAL4 is initially into a cassette. It's inactive, and it is in a cassette as like this one. This cassette has a promoter, in this case actin 5C, but it could be anything. Then it has an FRT sequence, and then it has terminator sequences, and inside of them it may have some marker, okay, some in this case it's yellow, but you can put a fluorescent protein here or whatever you want. Then another FRT sequence, and then the GAL4 gene. So the promoter cannot access it to the GAL4 gene, okay, in this circumstance. So GAL4 is inactive. So you have a strain with this cassette, and you cross it to a strain that has the UAS sequence upstream of a gene of interest. In this case it's GFP, but it could be the coding sequence of any gene or an RNAi or whatever you want. And this strain also has a cassette with the flypase enzyme, which also comes from yeast, and the heat shock promoter. The heat shock promoter is just a regulatory sequence that gets active when you put the fly at a high temperature. So after crossing both strains, you get flies that have all the cassettes on them, and if you give heat shocks to some of the flies, this heat shock promoter gets activated, and the flypase enzyme gets expressed, and it recognizes the FRT sites and removes these cassettes, which means that it moves the terminator sequences. So you get a cassette, an excised cassette, like the one at the left, where the actin promoter can actually activate transcription of GAL4. Then GAL4 protein is produced, and GAL4 binds to the UAS sequence, and it activates the expression of whatever gene is under this. So this heat shock promoter is not activated uniformly in all cells. Instead, it's very stochastic, and also the activity of the flypase is not super efficient. So the cassette is only excised in a few cells, and by regulating the intensity or the length of the heat shock, you can regulate how many clones or in how many cells the cassette is excised. As the cells grow, all the daughter cells of cells that have excised the cassette also carry the excised cassette, of course. So you get patches of cells like the ones in this figure that are expressing the gene that was under the UAS sequence. The position and identity of imaginal disc is determined mostly by Hox genes, which provide positional information in the embryo. So at embryonic stage 11, more or less, these thoracic segments, these thoracic primordia start to form in red, and later two different primordia, a ventral one and a dorsal one, are developed. At this stage, DPP is expressed in a horizontal band here, and DPP activates the identity of the dorsal primordia by activating genes such as snail, and at a mid concentration, DPP activates the identity of the ventral primordia by activating genes just as distantless. Also, at this stage, wingless is used to be expressed in a vertical band in a dorsoventral band here, and DPP also represses wingless. There's a third player, which is EGF, or vein, which is the ligand of EGFR, which is expressed in the most ventral part. And EGFR promotes the identity of the ventral primordium and represses the identity of the more dorsal primordium by repressing genes, just like snail. So this dorsal primordium, which is expressing snail, is the one that is going to give rise to the wing and to the halter, well, to the wing in the second thoracic segment and to the halter in the third. And the ventral primordium is the one that is going to give rise to the legs. Here you can see how these the early markers become expressed. In red, distalless, and in green you have snail. And you can see how snail becomes expressed in T2 and T3 during these embryonic stages. And then in the right figure you can see all the cells that are derived from this original primordial. You can see in green all the cells that have expressed snail before in previous stages, and in red all the cells that have expressed distalless in red. So as you can see, there's some mixing. There are a few red cells here and a few green cells here, but most of the wing and the halter are derived from the dorsal primordia, whereas most of the leg is derived from the ventral primordium. After determining during embryonic stages which cells are going to be part of the wing, during larval stage 2, it has to be determined which cells are going to be part of the notum, the hinge, and the blade. There are two main pathways that take part in this decision, and these are the EGF pathway, which is epidermal growth factor, which ligand is vein and is expressed in a gradient coming from the most dorsal part. And so vein binds to EGFR, which activates the expression of homothorax or HTH, THH, and the IROC complex of homeodomain genes. On the other hand, wingless, which is expressed in the most ventral part, represses vein and activates the expression of wing genes such as nubin and vestigial. Here you can see a regular fly, a wild-type fly. In red, the region where wingless is expressed, and then in green, the region where the EGF gradient is higher, which will form the notum. If wingless is mutated, this vein expression of the green part is expanded, and you don't have a pouch in the wing disc. And in the adult, instead of having a wing, you have like a second notum, or this part here, which is marked with an N prime symbol. On the other hand, if you have an EGFR mutant, the disc looks like that. The domain of wingless expression is much broader, and in the adult you get a fly that doesn't have a thorax body wall. You don't have a notum. Of course, wings don't develop properly, but because the effect is quite pleiotropic. And here you have the expression pattern of the genes. You have this wild type in dark blue vein expression and light blue wingless. If vein is mutated, wingless expression remains as before, but if wingless is mutated, vein expression expands towards the most ventral part. This is why you get this adult phenotype with a protruding second notum here instead of a wing. So this is what actually happens. In the beginning, in the early disc, both homothorax and TSH, which are notum selectors mostly, are expressed all over the disc, whereas when wingless starts expressing, it represses the effect of the vein through activation of other genes like nubin and vestigial. Indeed, if you look at this last picture, you have to notice that in the hinge there's there's wingless, but there's also homothorax. So whereas in the blade, wingless is repressing homothorax, in the hinge, wingless is not repressing homothorax, and this is because wingless represses homothorax through vestigial. Here you can see the same thing. This would be homothorax expression, and how it is progressively constrained to places where wingless is not expressed. And here you see how indeed in the wing pouch, wingless expression and wingless is green and homothorax is red. Okay, so in the pouch there's wingless and there's no homothorax, but in the border, in the region that's going to be to form the hinge, both of them are present. Then in E you can see expression of nubin, which is an important marker of the wing, and in F you can see vestigial. So here's a proof that vestigial is the one that is repressing homothorax. In this mutant clone for vestigial, homothorax in red is overexpressed, whereas if you overexpress vestigial in a clone in the more notum or hinge part, then homothorax is lost. And here the phenotype of mutating homothorax is shown. As you see, this is a wild-type hinge, and here's the hinge of a homothorax mutant. And as you see, its morphology is not normal. Then DPP is also relevant to repress some notum genes. So as you see here in the A and B figures, in red you have the expression pattern of DPP. So these expression patterns are mostly exclusive. So remember that DPP is a morphogen, so these cells are also receiving DPP signaling. And when you overexpress some DPP here in the notum part, IROC genes are repressed.